So, I mean, it's more for the audience to ask the questions in this particular panel, but I'd like to start off. First, by saying thank you very much for brilliant presentations. Thank you. I'd like to ask a question to both Paulo and Jim. Firstly, in terms of, Jim, your system, obviously you're looking at scaling this out. You're working in Africa. You mentioned Cameroon as well. I just wanted to pick up on the data generated. You showed a lot of data that was generated. Is there a system in place to house that data? We heard earlier about weak data management. What's going to happen with the data generated by Muse? I suppose the same question to you, Paulo. Is that factored into the way that you're progressing your system? So firstly to Jim.
Well, as we go through the regulatory process, that data is preserved, and it goes into our clinical file, which is examined by the regulatory groups that monitor us. And initially, the product will be released for research only. So, but the other data will be stored, and will generate additional data to go for an FDA 510K claim.
Okay, and are you part of that storage, or is that more left to the government's?
Right.
As an example,
we have a regulatory body that reviews it internally and externally.
Okay, excellent. So it's robust. And the same answer you, Paulo, in terms of you're developing your tool. You mentioned you're looking for funding. It's going to be generating a lot of data, differential data between the chikungunya, dengue, different viruses. Are you planning to house that data, to integrate to that, to offer something?
The idea is start with a small pilot. Proof of concept is already done, but in Brazil, especially with dengue, we are going to partner with the healthcare system there. Then collect data and see how it works and monitor first as academic. Then after start a broad approach, but this will be made by, we will try to do small, but already collecting by a database where it can be controlled in terms of the database. So the idea, but let's see how this grows.
And just building on something that Lisette had said, you mentioned there's a feasibility for a worldwide ecosystem. You've shown that. You've got 30 companies or participants in the Netherlands, where you're in the region that you're based. What are the challenges? Have you had challenges getting external people to participate in this? And what are those challenges?
Yes, maybe I should clarify a little bit that the 30 participants is for the protozoa, and of these 30, I think about 5 of them are not in the Netherlands. Yeah. So we even have some participants from Canada and also from other European countries. The reason for the protozoa that we're more focusing on the Netherlands is because they're completely unpreserved samples, stool samples, because we want to be as real life as possible. For the helminths, we preserve them in ethanol, and in principle it can be distributed to everywhere. We also give a much larger time window for participants to reply. So normally it's about 3 or 4 weeks for the protozoa, but for the helminths we did several months. So there is more time also for the shipment and for actually performing the PCR. So I think the major limiting factor at the moment is to have enough material, to have enough good validated material, really, we need pots of stool. That's actually
okay, good answer. Would there be questions from the floor to ask? I think there's a Poppy at the back.
Hi, I'm Poppy Lamberton from the University of Glasgow. Thank you. I actually just wanted to ask you, Lisette, a question about the educational samples that are very, very low infection intensity. And how many repeats does each laboratory do? And do you see more inter-laboratory variation than you do intra-variation? And how might you be able to use these data to maybe produce some kind of estimates of error for the future for low intensity on a larger sort of sample size?
These educational samples, what is happening is that very often, it's not the 5 flow is often the CQ, the difference in CQ value is within the limitations. That's not a problem. But what you see is that just some laboratories are not able to be as extremely sensitive as others. Laboratories they receive only one single sample participant. So they can, if they want, they can repeat and try if for example, if their CQ value is really around 38 for those who are familiar with it. And they do it 5 times, and they might see that it's 2 times positive and 3 times negative. But if they think that the positive result is truly positive, then their hand in the result as being positive. That's up to them. So the participants again don't receive 5 flow of the same sample, only the reference centers. And of course it gives you information, because if you know as a participant that every time when there is an educational sample you're able to detect it, then that means that your assay is more sensitive than maybe from another laboratory who's able only to detect 50% of these educational samples.
I have a question for Lisa. I mean, you mentioned LF, the disease setting, but you mentioned you can pick up other pathogens, and not the funding is an issue, but it's something to look at. I mean, would there be any kind of reach out message to other communities, disease settings outside of the one that you've worked in using your approach?
So, our first setting was in Ghana because we were targeting filariasis. We have also conducted a study in India, and so we're running the lab samples now. That would be for picking up Leishmania parasites. And I think that the benefit of the tool is related to how difficult it is to detect. So the team that we were working with in India, for example, they're collecting sandflies and Culex mosquitoes using a light trap, and they're throwing away, sort of tenfold the number of mosquitoes in order to find those few sandflies. And I think that if this method works in a setting like that, then I think that there would be scope to roll that out. We're also going to be going to American Samoa, where there's potential for Ross River virus. So we also need to look at arboviruses. And I think that, again, the utility is also going to relate to how do we respond when we find positives. And I think that there will really be scope there for the arboviruses, because if we find positive mosquitoes, then that tells us where transmission from humans to mosquitoes is happening. Because I think oftentimes we see where does a positive human turn up, and then we spray around their home, but that isn't necessarily where they're infecting mosquitoes. So I think this is maybe a long answer to your question, but in terms of where we would go, I think we need to think about what is sensible to multiplex within a given setting, and that's going to be very different, based on the geographical location, as well as whether we want to use this tool to detect sort of emerging arboviruses, or whether we want to use it to detect and monitor low levels of these eliminable diseases. So I think that we would want to partner with anybody who is doing surveillance for one disease and think about, okay, you can have your samples that you're collecting, but give me what's at the bottom of that cup, and I can screen it for a lot of other things. So.
That's quite a strong message for partnership. Would there be any other questions from the floor on that particular issue, the multiplex issue? Total silence. Yeah, it's always good. Always good. Yeah. Is that a wonderful part of diagnostics? There's no answer. There's no questions for. Thank you. Yeah.
So, Lisette, this is about PCR technology. Would that work for LAMP also? That's the first question. And then the second one that I want to ask is, so you express it as CQ values, but are you going to standardize this in genome equivalence programs too, or something like that?
Two very relevant questions. Yes, you can also participate if you do LAMP technology, and even if you do it non-quantitative, because you can the outcome actually is qualitative. But by providing the CQ value, you give additional information for us as an organization. Because we take the average of all CQ values, all participants, and then again you have the information whether you always score below or above the average. But it's not obligatory. You can do a LAMP or any other qualitative test. Your second question was about
Genome equivalence.
Oh yeah, now again, because not all the laboratories use the this kind of standardization by targets per ml or so on. Most laboratories use CQ or CT value as an output, so that's why we use it for the moment. I don't think, as long as it's not standardly used, we won't change it into targets per ml or any that kind of outcome.
Thanks, Lisette. I think one more question for Jim. Outside of malaria, are there other areas you're working on in terms of applying that approach, Muse approach to?
The Global Health Institute is working on 3 different projects. With every project, we use a holistic approach. We develop pharmaceutical applications, diagnostic applications. We work on medical devices. Whenever we take on a disease, we try and use all these approaches. And currently, we're working on malaria, schistosomiasis, and antimicrobial resistance.
Okay, brilliant. Thank you. Any other questions from the floor for the panellists at all? If not, then we can break for coffee now. So a round of applause to the panellists.